Question number 46 is from the chapter Photosynthesis in Higher Plants, class 11, and the question is, which of the following statements is incorrect? Option A, both ATP and NADPH plus H plus are synthesized during non-cyclic photophosphorylation. Option B, stroma lamellae have PS1 only and lack NADP reductase enzyme. Option C, grana lamellae have both PS1 and PS2. And option D, cyclic photophosphorylation involves both PS1 and PS2. So the incorrect statement is statement D, because cyclic photophosphorylation involves only PS1 and not PS2. Okay, rest options are correct. Let's see how. We know that photophosphorylation is the process of ATP formation from ADP and inorganic phosphate, right? Now this ATP is synthesized by the plants using the radiant energy absorbed during the photosynthesis, okay? Now, photophosphorylation can be of two types. It can be a non-cyclic photophosphorylation or it can be a cyclic photophosphorylation. So the first difference is the location, that non-cyclic photophosphorylation takes place in the thylakoid membrane or grana lamellae of the chloroplast. Okay. Whereas cyclic photophosphorylation takes place in the stroma lamellae of the chloroplast. Right. Then the major difference is that in case of non-cyclic photophosphorylation, two photosystems are functional, PS1 and PS2, and both the photosystems work in series, first PS2 and then PS1. And each photosystem consists of a light harvesting complex and an electron acceptor. Okay. So one light harvesting complex and one electron acceptor in PS1 and one light harvesting complex and one electron acceptor in PS2. Then both the photosystems are connected through an electron transport system. So electrons will be transported from one photosystem to the other. So here we can see that option C is a correct statement because grana lamellae contains both PS1 and PS2. Okay. Then, while getting transported from one PS to second PS, the electron is responsible for the formation of ATP molecule by transferring the inorganic phosphate to the ADP molecule. Okay, so one ATP will be formed here. Then, after passing through both the photosystems, the electron is received by an energy-rich NADP molecule, which will get reduced to NADPH plus H plus with the help of an enzyme, NADP reductase enzyme. Okay, that means grana lamellae contains NADP reductase enzyme. So here we can see option A is also correct because it says that both ATP and NADPH and H plus are synthesized. So we saw that both the things are synthesized here. Okay, now what happens in case of cyclic? Here only PS1 is functional and not PS2. Okay. Also, there is no NADP reductase enzyme in the stroma lamellae. Therefore, no NADPH is formed. Okay. So, option B is also correct because it has PS1 only and it lacks NADP reductase enzyme. So, the only incorrect option we found out is option D. So, it will be the correct option here. I hope you understood this. Question number 47 is from the chapter Biological Classification, class 11, and the question is, which of the following statements is correct? Option A, fusion of two cells is called karyogamy. Option B, fusion of protoplasms between two motile or non-motile gametes is called plasmogamy. Option C, organisms that depend on living plants are called saprophytes. And option D, some of the organisms can fix atmospheric nitrogen in specialized cells called sheath cells. So the only correct statement here is statement B and rest of these statements are incorrect. Okay. So let's see, we know that the reproduction in fungi can take place in two modes. It can either be in asexual mode or through sexual mode. Okay. The asexual reproduction in fungi involves the processes such as fragmentation, fission or budding. Whereas the sexual mode of reproduction in fungi involves three steps. First one is plasmogamy, second one is karyogamy, and the last step is meiosis in the zygote. Okay. What is plasmogamy? It is the fusion of protoplasms. Okay. Protoplasm of the gametes. Okay. Whereas in the next step, the nuclei are fused. So the fusion of nuclei of the gametes is known as karyogamy. Therefore, option A is incorrect because it is not the fusion of cells but nuclei. And option B is correct. What does option C says? Organisms that depend on living plants are called saprophytes. So this is also incorrect because saprophytes grow on dead matters. Okay. So dead material, whereas the organisms which grow on living organisms are known as parasites. Okay. What is option D? Some of the organisms can fix atmospheric nitrogen in specialized cells. So these cells are known as heterocyst and not the sheath cells. Okay. And these heterocysts are present in some blue-green algae. Okay. So I hope you understood this. Question number 48 is from the chapter Biomolecules, class 11, and here we have to match list 1 with the correct option in list 2. So the first one is protein. What is protein? We know that it is a polypeptide. This means that it consists of more than one peptide bonds. Therefore, the correct match for protein will be peptide bonds. Okay, what is peptide bond? It links to amino acid, and more specifically the carboxyl group of one amino acid to the amino group of next amino acid. Okay, so CO from here and NH from here combine to form CONH, and this is the peptide bond. Okay, and the remaining OH from here and one hydrogen from here combine to form water molecule, which is removed. So it leads to the removal of water molecules. Okay. Now the next one is unsaturated fatty acid. So the correct option for this will be number 1, that is carbon-carbon double bonds. Saturated will contain carbon-carbon single bond and unsaturated will contain double bond. Okay. The next is nucleic acid, and the correct match for this is phosphodiester bonds. So this bond links to nucleotides. Okay. Now here one phosphate group reacts with hydroxyl group, and this leads to the formation of ester bond. Now since two such ester bonds are present, one on either side, therefore it is known as phosphodiester bonds. That means two ester bonds are there. Okay. Now the last one is polysaccharide. So polysaccharides contain glycosidic bonds. Okay. So the correct option out of the given 4 will be option A, right? So I hope you understood this. Question number 49 is from the chapter Molecular Basis of Inheritance, class 12, and the question is DNA fingerprinting involves identifying differences in some specific regions in DNA sequence called as satellite DNA, repetitive DNA, single nucleotides, and polymorphic DNA. So the correct option is option B, that is repetitive DNA. So what are repetitive DNA? These are DNA sequence where a small stretch of DNA is repeated many times. Okay, and these repetitive DNA are separated from the bulk DNA on the basis of density through a process known as density gradient centrifugation. Okay, so here the bulk DNA appears as a major peak. Okay, whereas the smaller peaks are referred to as satellite DNA. So you understood what is satellite DNA, and one of the characteristics of satellite DNA is that they show high degree of polymorphism. Okay, what is polymorphism? It is the variation at genetic level. Therefore, we say that the basis of DNA fingerprinting is VNTR. What is VNTR? It means variable number of tandem repeats. And what is variable number of tandem repeats? It is a satellite DNA which is used as probe that show high degree of polymorphism. Okay, so I hope you understood this. Question number 50 is from the chapter Mineral Nutrition, class 11, and here we have to match the name of bacteria mentioned in column 1 with the process they carry out mentioned in column 2. So these processes are related to nitrogen cycle. And we know that nitrogen is one of the most important nutrients for natural as well as agricultural ecosystem, and it exists as two nitrogen atoms joined by very strong triple covalent bond. Now this nitrogen has to be fixed in our environment. There are many ways by which this can be fixed, but one of them is biological nitrogen fixation, which we have to deal with. Okay. So what happens during biological nitrogen fixation? First, nitrogen is converted into ammonia, and this process is known as nitrogen fixation. One of the bacteria that is involved in this is rhizobium. Okay. So the correct match for rhizobium will be conversion of atmospheric nitrogen to ammonia. Now after that, this ammonia has to be oxidized into nitrogen oxides, and this process is known as nitrification. Okay. This nitrification takes place in two steps. In the first step, ammonia is oxidized into nitrite, that is NO2 minus. Okay. So some of the bacteria involved in this is Nitrosomonas and Nitrococcus. Okay. So you will see here Nitrococcus is mentioned. So what will be the process here? Conversion of ammonia into nitrite. Okay. In the next step, nitrite is converted into nitrate, that is NO3 minus, and the bacteria involved in this process is Nitrobacter. Okay. So the correct match for Nitrobacter will be conversion of nitrite to nitrate. Understood? Okay. Now this nitrate will be absorbed by the plants and will get transported to leaves. Okay. Now there is also a reverse process by which the nitrate present in the soil is converted back into nitrogen. Okay. And this is known as denitrification. Right? Some of the bacteria that are involved here are Pseudomonas and Thiobacillus. So the correct match for Thiobacillus will be denitrification. Got it? I hope you understood this. So the correct option out of the given four will be option A. So question number 51 is from the chapter Biotechnology Principles and Processes, and the topic is Tools of Recombinant DNA Technology. Okay, so the question says a specific recognition sequence identified by endonucleases to make cuts at specific locations within the DNA is, and the options are degenerate primer sequence, Okazaki sequences, palindromic nucleotide sequences, and polyadenylate tail sequences. So the correct option out of the given four is option C, that is palindromic nucleotide sequences. Okay. So we know that in recombinant DNA technology, a cut is made within the DNA at specific location. Now this location is identified by restriction endonuclease. Okay. And what type of location is identified? A palindromic site or a palindromic sequence. Okay, so it is basically a sequence of the nucleotides where complementary strands of DNA read the same in both direction. Okay, for example, this is one of the strands 5' to 3'. This is the next strand 3 dash to 5 dash. Okay. So now if we have to read the sequences, suppose from 5 dash to 3 dash, and it reads A, T, G, C, then the next strand will also read in the same way from 5 dash to 3 dash. That means A, T, G, C. Do you understand that? A, T, G, C from 5 dash to 3 dash. Similarly here also 5 dash to 3 dash will be in this manner. Okay, that means A, T, GC. So this type of sequence is known as palindromic sequence. Okay. Now let's see the other options. Option number A says degenerate primer sequence. So what is degenerate? See, a PCR primer sequence is called degenerate if some of its positions have several possible bases. Okay. So if there are several possible bases, then it will be said to be degenerate primer sequences. Okay. Now, what is Okazaki sequence? These fragments are synthesized discontinuously during DNA replication. Okay, suppose replication takes place. So one of the strands is synthesized in a continuous manner, whereas the other is synthesized in discontinuous manner. Okay, so these are known as Okazaki fragments. And then later on these are joined with the help of DNA ligase. Okay, so these will be later on joined. Okay, now what is polyadenylate tail sequences? Polyadenylate tail sequences basically consists of a large number of about 200 to 300 adenylate residues. Okay, therefore these are known as polyadenylate tail. Okay, so these residues are added at 3' end of hnRNA. Okay, during the post-translational modification. During post-translational modification in which type of organisms? Eukaryotes. Okay, and not in prokaryotes. Okay, so question number 52 is from the chapter Cell Cycle and Cell Division, class 11. And the question says, the fruit fly has 8 chromosomes in each cell. During interphase of mitosis, if the number of chromosomes at G1 phase is 8, then what would be the number of chromosomes after S phase? Okay, and the options are 8, 16, 4 and 32. Okay, so this is very simple question. We just have to understand that what happens during S phase. Okay, during S phase, the most important thing that happens is that DNA is duplicated. What does this mean? That the DNA or the amount of DNA will increase, or you can say doubled, but the number or amount of chromosomes remain same. That means the number of chromosomes is not affected. Okay, so it is very simple that if the number of chromosomes at G1 is 8, it will remain 8 even after the S phase. Okay, so the correct option will be 8. Okay. Okay, so let's come to question number 53. It says which one of the following belongs to the family Muscidae. So we have several insects mentioned here: firefly, grasshopper, cockroach and housefly. So we have to tell which one of them belongs to the family Muscidae. Okay, but before that, just remember that all of them are insects, so they belong to the class Insecta. Okay, so their class is same but family is different. Now, Muscidae is the family that housefly belongs to. So, option D will be the correct option for this. but we'll also see the family of other insects mentioned here. What is the family of firefly? It is Lampyridae. Okay, that means the fireflies belong to the family Lampyridae. Which family does grasshopper belongs to? It belongs to Acridae. And cockroach belongs to a family called Blattidae family. Okay. So you have to remember the names of families and the insects that belong to them. Okay. So now come to question number 54. It is from the chapter Digestion and Absorption. Okay. So the question says the succus entericus is referred to as, and the options are pancreatic juice, intestinal juice, gastric juice and chyme. So the correct option is option B, that is intestinal juice. Okay. That means the juice secreted by intestine is known as succus entericus. Right. Now, this was a very direct question. We'll also deal with other options. First one is pancreatic juice. Okay. So as the name suggests, you can easily guess that it is a juice that is released by the pancreas. Okay. But we know that pancreas have two types of secretions. First is endocrine secretion and the other is exocrine secretion. Right. So out of them, which type of secretion involves pancreatic juice? It is exocrine secretion. Okay. This is known as pancreatic juice. So we can write here exocrine secretion of pancreas. Now the last two options are gastric juice and chyme. So both are released in the stomach. Okay. But the difference is that gastric juice is secretion of the gastric glands. Okay. Okay, and these glands are present in the stomach. So both are present in stomach. So you can remember this. The only difference is that the gastric juice is released from the gastric glands, whereas chyme is referred to the acidic food. Okay, that is present in the stomach. Okay, so I hope you understood this. Okay, so the next one is question number 55, which is from the chapter Biotechnology and its applications. And the question is, with regard to insulin, choose the correct options. Now, before we have a look at the options, let's try to understand the structure of an insulin. Okay. So an insulin mainly consists of two chains of polypeptides, chain A and chain B. Both are polypeptide chains. Okay. And these two chains are connected through disulfide bridges. Disulfide bridges means containing two sulfide bonds. You can see here. Therefore, these are known as disulfide bridges. Okay. Now, when an insulin is formed initially, it is formed as a proinsulin and not as matured insulin. Okay. So the proinsulin consists of an extra stretch of peptide which is known as C-peptide. But later on, as it gets matured, this C-peptide is removed or separated from the main insulin, and what is left? Only chain A and chain B connected by disulfide bridges. Okay. So this is the mature insulin. Now let's see what the options say. Okay. So statement A is C-peptide is not present in the mature insulin. So this is a correct statement. We saw in mature insulin C-peptide was separated, right? Option B is the insulin produced by rDNA technology has C-peptide. So what kind of insulin is produced by rDNA technology? Mature insulin. Okay. So mature insulin is produced using rDNA technology. So we can see here that mature insulin does not contain a C-peptide chain. Therefore, this is a wrong statement. Okay. Statement C is the proinsulin has C peptide. So this is true. We saw here now that the proinsulin had a C peptide chain attached with it. Now the last option is A peptide and B peptide of insulin are interconnected by disulfide bridges. So this is also true. We have already seen. Okay. So how many options are correct? A, C and D. So out of the given options, option C is correct. Okay. I hope you understood this. So moving on to question number 56, which is from the chapter Body Fluids and Circulation. And the question is, persons with AB blood group are called universal recipients. This is due to absence of antigens A and B on the surface of RBC, absence of antigens A and B in plasma, presence of antibodies anti-A and anti-B on RBCs, and absence of antibodies anti-A and anti-B in plasma. Okay. So here you will see two terms are used, antigen and antibody, right? Also, two different locations are mentioned here, the surface of RBC and plasma. So let's see what are antigens and antibodies and where are they located. Okay. So an antigen is generally any foreign substance to which our antibody can respond. Okay. To which antibody can respond. So here you can understand that the role of antibody is to respond to the antigens and fight them off. Okay. So these are produced by our immune system. This is the most important thing. This is how our immune system protects us from various diseases, because the antibodies produced by them fight with any foreign substance, or we can say they fight with antigens. Okay. And what is their location? Antigens are found on the surface of RBCs, whereas antibodies are found in the blood plasma. Okay. Now, different blood groups contain different antigens and antibodies. Okay. So let's see. What are the major blood groups? There are four types of blood groups. Okay. These are A, B, AB and O. Now, the name of the blood group depends on the type of antigen present in it. For example, the blood group A contains the antigen A, whereas the antibody present in it is anti-B. That means people having blood group A cannot receive blood from people having blood group B. Otherwise, this antibody will react to it and fight it off. Okay. Similarly, blood group B has antigen B and the antibody is anti-A. So they cannot receive blood from people having blood group A. What about AB? Here both the antigens are present and no antibody is present. Therefore, there is nothing to fight. Okay. So they can receive blood from people having any blood group, and therefore they are known as universal recipient. Okay. So this was the answer. Let's see about blood group O. There is no antigen, but both the antibodies are there. That means they cannot receive blood from anyone except having blood group O, but they can give blood to anyone. So they are known as universal donor. Okay. But here, according to question, we have to answer about AB. Since they don't have antibodies in their plasma, therefore option D will be correct. That absence of antibodies anti-A and anti-B in plasma is responsible for them being universal recipient. Okay. Now come to question number 57, which is from the chapter Principles of Inheritance and Variation, class 12. And the question is, in a cross between a male and a female, both heterozygous for sickle cell anemia gene, what percentage of the progeny will be diseased? And the options are 50%, 75%, 25% and 100%. Okay, so let's first understand what is sickle cell anemia and under what condition the disease shows up. So just remember that sickle cell anemia is an autosome linked recessive trait. Okay, recessive trait. And the disease is transmitted from parents to offsprings only when both the partners are carrier for the gene. So here, according to this question, the male and female both are carrier for the gene. That means both are heterozygous. Okay. So the disease will be transmitted to all the offsprings. Okay. So if we see that the pair of allele that is responsible for controlling the disease is HbA and HbS. Okay. This single pair of allele controls the disease. Okay. What is the possible genotype of these alleles? It can be HbA, 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 HbS, again HbA, HbS or HbS, HbA, both are same. And then the last one is HbS, HbS. So these are different combinations that are possible. So these are the possible genotypes. Okay. So will all the genotypes show the diseased phenotype? No. Only the last one, that is HbS, HbS, will show the diseased phenotype. And the rest will apparently be unaffected, but they will be the carrier of the disease. Okay. These are just carrier, but these won't appear as the diseased one. Okay. Now, if a cross between male and female is carried out and both having HbA, HbS, so what kind of springs we will get? Similar, right? That means HbA, HbA, then HbA, HbS, then again HbS, HbA, or both are same. Okay. Then HbS, HbS. Okay. So which one will show the diseased phenotype? Only the HbS, HbS. Okay. That means 1 out of 4 possible progenies will show the disease phenotype. And what will be the percentage? 1 by 4 multiplied by 100, and it will be 25%. So the correct option will be option C, that is 25%. Okay. I hope you understood this. So the next one is from the chapter Body Fluids and Circulation, and the question is, which enzyme is responsible for the conversion of inactive fibrinogens to fibrins? And the options are thrombin, renin, epinephrine and thrombokinase. So the correct option is option A, that is thrombin. Okay, so this question is based on the concept of coagulation of blood. Okay, so what happens during coagulation? A coagulum or a clot is formed which prevents the excessive loss of blood from the body. Okay, excessive loss of blood. Now, this clot or coagulum consists of thread-like structures known as fibrins. And what is the role of fibrins? Dead and damaged particles are trapped in these fibrins. Okay. Now, let's see how these fibrins are formed. So, the process of formation of fibrins consists of mainly 3 steps. In the first step, an enzyme complex thrombokinase is formed. Okay, and this is formed by a series of enzyme linked reactions, and this series is known as cascade process. Okay, so this is the first product here. Now, in the second step, this thrombokinase catalyzes an important reaction. What does it do? It helps in the conversion of inactive prothrombin into an active thrombin. Okay. So who does this work? Thrombokinase, right? Now, the last step is the role of this thrombin. So this thrombin helps in the conversion of inactive fibrinogen into active fibrin. Okay, so according to this question, this conversion of fibrinogen into fibrin is done by thrombin. Therefore, option A is correct. Okay. Now, what does the next option say? Next is renin. Okay. So, renin is actually secreted by juxtaglomerular cells of the kidneys in response to glomerular blood flow, which converts angiotensinogen into angiotensin 1. Okay. And what is epinephrine? Epinephrine is also known as adrenaline, and it is secreted by adrenal medulla. Okay, in response to the stress of any kind or during any kind of emergency. Okay, so I hope you understood this. Now we will see question number 59, which is from the chapter Breathing and Exchange of Gases. Okay, and the question is the partial pressures in millimeter of mercury of oxygen and carbon dioxide at alveoli. Okay, alveoli is the site of diffusion. So we have to determine the partial pressures of oxygen and carbon dioxide at the alveoli here. Okay, so we have different options here. See, these partial pressures vary in different conditions. Okay. For example, in atmosphere, the partial pressure of oxygen is 159 and that of carbon dioxide is 0.3. Okay. So this option D is the partial pressure of oxygen and carbon dioxide in the atmosphere. Okay. Then in blood also it varies, and also on the type of blood. For example, in deoxygenated blood, the partial pressure of oxygen is 40 and partial pressure of carbon dioxide is 45. So this is for the deoxygenated blood. On the other hand, in case of oxygenated blood, the partial pressure of oxygen is 95, whereas that of carbon dioxide is 40. Okay, so option C is for oxygenated blood, right? And the most importantly, that is asked in the question, the partial pressure of oxygen at alveoli is 104, whereas the partial pressure of carbon dioxide is 40. So this is at alveoli. Okay. Therefore, option A will be the correct option for the partial pressures of oxygen and carbon dioxide at alveoli or the site of diffusion. Okay. Now come to question number 60, which is from the chapter Locomotion and Movement, class 11. And the question says, chronic autoimmune disorder affecting neuromuscular junction leading to fatigue, weakening and paralysis of skeletal muscle is called as arthritis, muscular dystrophy, myasthenia gravis or gout. So the correct option is option C, that is myasthenia gravis. It is a chronic autoimmune disorder and it affects the neuromuscular junction, which leads to various problems such as fatigue, weakening and paralysis of skeletal muscles. Okay. Now next, what options are there? Option A is arthritis. Okay. What is arthritis? It is generally the name of inflammation in the joints. Okay. Inflammation in the joints, and it is one of the very common problems nowadays, isn't it? The next one is muscular dystrophy. It is a genetic disorder, right, which results in progressive degeneration of the skeletal muscle. Okay, so it causes degeneration of skeletal muscle. And the last option was gout. Okay, what is gout? It is a disease which is caused due to deposition of uric acid. Okay, so uric acid crystals in joints and leads to its inflammation. Okay, hope you understood this. Question number 61 is from the chapter Molecular Basis of Inheritance, class 12. And the question is, which is the only enzyme that has the capability to catalyze initiation, elongation, and termination in the process of transcription in prokaryotes? Okay. And the options are DNA-dependent DNA polymerase, DNA-dependent RNA polymerase, DNA ligase, and DNase. Okay, so the correct option out of the given four is option B, that is DNA dependent RNA polymerase. Okay, so we know that the process of transcription involves copying of genetic information from one strand of DNA into an mRNA. Okay, and the enzyme used is RNA polymerase generally, but there is some variation in case of prokaryotes and eukaryotes. Okay, the process in prokaryotes is quite simple. So only a single enzyme known as DNA dependent RNA polymerase enzyme is used for the all the steps. Okay, that is initiation, elongation, and termination. Okay, so this is our answer. Whereas what happens in eukaryotes, there are certain complexities. Therefore, different polymerase enzymes are used for all these steps. Okay, these are RNA polymerase 1, RNA polymerase 2, and RNA polymerase 3. Okay, so we know why DNA dependent RNA polymerase is the correct option here. Okay, what are the other options? Option A is DNA dependent DNA polymerase. This is an enzyme which is used during the process of DNA replication. Okay, what happens during DNA replication? We know that the genetic information is copied from one strand of DNA into a new strand. Okay, that means the DNA is duplicated. Also, we know that during this process, one of the strands is synthesized in a continuous manner, whereas the other strand is synthesized in a discontinuous manner, right? As you can see here. So these strands, these broken fragments are connected or joined using an enzyme known as DNA ligase. Okay, so this DNA ligase is used as a glue, okay, to join the discontinuous DNA fragments. Okay, now the last one is DNase. What is DNase? It is basically an enzyme that helps in degrading the DNA. Okay, if we have to break down the DNA, then we have to use DNase. Okay, so it degrades the DNA. Hope it is clear to you. So now question number 62 is from the chapter Molecular Basis of Inheritance, class 12. And the question is, which of the following RNAs is not required for the synthesis of protein? So what is the method that is involved in the synthesis of protein? It is translation, right? Now, the options are mRNA, tRNA, rRNA, and siRNA. We know that mRNA, tRNA, and rRNA all are involved in the translation. Therefore, the only RNA which is not involved is option D, that is siRNA. What does siRNA mean? It means small interfering RNA. Okay. There are other names as well. Sometimes it is also known as short interfering RNA or silencing RNA. And it is a class of double stranded RNA. Okay. We know that RNA can be double stranded as well as single stranded, right? So this is one of the double stranded one. And it typically consists of 20 to 24 base pairs, right? It operates within the RNA interference pathway. Okay, so it has role in RNA interference pathway, but not in the translation, right? Now let's see the full form of mRNA, tRNA, and rRNA. We know M here stands for messenger. So this is the messenger RNA. tRNA is transfer RNA. Whereas rRNA means ribosomal RNA. And their role also related to their names. Okay, what does messenger do? Messenger carries information, isn't it? Therefore here the mRNA is the RNA that carries the genetic information that is provided by the DNA. Okay, so suppose this is the mRNA and there are all the information, that means the bases, nucleotides. Okay, what does tRNA do? As it you can see here, it is transfer RNA. So it helps in transferring the amino acids on the mRNA. Okay. So suppose this is tRNA. So it will keep adding the amino acids here. Okay. One by one. And what does ribosomal RNA do? It is a structural RNA and it forms ribosomes which are involved in the translation. Okay. So ribosome, we know ribosome has two subunits, one small subunit and a large subunit. Okay, so this is the ribosome. It is formed by ribosomal RNA. Okay, so I hope you understood this. Now next one is from the chapter reproductive health, and the question is, which one of the following is an example of hormone releasing IUDs? Options are copper T, LNG20, copper 7 and multi-load 375, and the correct option is option B, that is LNG20. So let's first understand what are IUDs. IUDs stand for intrauterine devices. So as the name suggests, these are some of the devices which are inserted inside the uterus. Okay, so these are generally inserted by doctors or expert nurses in the uterus through vagina, and these are one of the effective and popular methods used today for birth control. Okay, these are presently available in three forms, or you can say three types of IUDs are there. Okay, first one is non-medicated IUDs. Second one is copper releasing IUDs. And the third one is hormone releasing IUDs. Okay. What are some of the examples here? The example of non-medicated IUDs is Lipis loop. Okay. Lipis loop. Examples of copper releasing IUDs are copper T, copper 7 and multi-load 375. Okay, and the example of hormone releasing IUDs is LNG20 and progestasert. Okay, so as asked in the question, the example of hormone releasing IUD as mentioned here is LNG20. So option B is the correct option, and rest all are copper releasing IUDs. Okay, I hope you understood this. Okay, so question number 64 is from the chapter Molecular Basis of Inheritance, class 12. And the question is, if adenine makes 30% of the DNA molecule, what will be the percentage of thymine, guanine and cytosine in it? Okay, and here are four possible options. Now, to solve this, we just have to know about a simple rule here, that is Chargaff's rule. Okay, what does this rule state? It states that the amount of adenine is equal to the amount of thymine in any double-stranded DNA. Okay. Similarly, the amount of guanine is equal to the amount of cytosine. Okay. Now here what is given? The amount of adenine is 30%. So you can say that the amount of thymine will also be 30%. Right? Now look at the options here. There is only one option in which the amount of thymine is mentioned as 30. So it is quite evident from here that option C will be the correct answer. but still we'll try to find out the amount of guanine and cytosine as well. See, we know that the total amount of adenine, thymine, guanine and cytosine will be equal to 100%, because these are the things which make up one DNA molecule, right? So here we know that the amount of adenine is 30, thymine is also 30, and all will be in percentage. The amount of guanine is unknown here, right? So let's assume it to be X%. Then if the amount of guanine is X%, what will be the amount of cytosine? It will also be equal to X%, right? Will be equal to 100. Now 30 plus 30 is 60, and X plus X is 2X equals to 100. 2X will be equal to 100 minus 60, that is 40. Okay, then what will be the amount of X? It will be 40 divided by 2, or 20. That means the amount of guanine will be 20%, and therefore the amount of cytosine will also be equal to 20%. Okay, I hope you understood this. Okay, so question number 65 is from the chapter Microbes in Human Welfare, class 12. And here we have to match list 1 containing the names of some important microorganisms with the correct option in list 2 having the names of products which are produced by the microorganisms. Okay, so the first one is Aspergillus niger. So what kind of microorganism is an Aspergillus niger? It is a type of fungus, right? And it is responsible for the production of citric acid. Which vitamin is found in citric acid? Vitamin C, isn't it? So the correct match for number A is 3. So we can write it here. The correct match for A is number 3. Okay. Now, the next one is Acetobacter aceti. Now, can you guess what kind of microorganism is this? Here you will see the word bacter, which is related to bacteria. Okay, so this is a type of bacteria, and it is responsible for the production of acetic acid. Okay, again you can see the similarity between aceto and acetic, so it is easy to remember the names. Okay, so the correct match for B will be number 1. Now the third one is Clostridium botulinum. Okay, now this is also a type of bacteria, right? And it is responsible for the production of butyric acid. Here again botulinum and butyric acid are similar, so again you can easily remember this. Okay, so this is butyric acid. What will be the correct match for C? It will be 4. Okay, and then the last one is lactobacillus. Again here it is written bacillus, so it's quite clear that this is a type of bacteria. Okay, and it is responsible for the production of lacto and lactic are similar, so lactic acid. So the correct match for D is number 2. So which of the given options correctly fit this? Number A, right? So this is the right option. Okay, so question number 66 is from the chapter Animal Kingdom, class 11, and the question is, read the following statements. First one is metagenesis is observed in helminths. So this is a wrong statement. Why? Because metagenesis is observed in the members of which phylum? Coelenterata. And the coelenterates are also known as cnidarians, and the phylum is Cnidaria. Why? Because of the presence of special type of cells known as cnidoblasts or cnidocytes. Okay. Now these coelentrates are generally found in two basic forms. First one is polyp and the second one is medusa form. Okay. So the polyp form is sessile, whereas the medusa form is free swimming. Okay. Other than that, there is a difference in shape. For example, polyps are cylindrical organisms. So they look something like this. Whereas medusa are umbrella shaped. Okay. like this. And the examples of polyp include hydra, adamsia, etc. Whereas medusa include aurelia or jellyfish. So as you can see, some exhibit polyp form, some exhibit medusa form, but there are certain coelentrates, for example obelia, which exhibits both the forms. So in that case, polyp produces medusa asexually, whereas medusa produce polyp sexually. Okay, so this is a kind of alternation of generation. So this alternation of generation is known as metagenesis. Got it? Now the second statement says echinoderms are triploblastic and coelomate animals. So what is the meaning of triploblastic? Triploblastic means having three embryonic layers. Okay, so we know that basically there are two embryonic layers. The inner one is endoderm, whereas the outer one is ectoderm. Okay, now in certain organisms there is an additional third layer in between the endoderm and the ectoderm. Okay, and this is known as mesoderm. So those organisms having only two endo and ectoderm are known as diploblastic, whereas those organisms having all the three layers are known as triploblastic. Okay, so the echinoderms are triploblastic. And what do you mean by coelomate? Coelom means body cavity, okay, which is lined by the mesoderm. Okay, so those organisms in which the body cavity is lined by the mesoderm are known as coelomates. So statement B is correct because echinoderms are triploblastic as well as coelomate animals. Okay. Now the third one is roundworms have organ system level of body organization. So this is also a correct statement. Okay. Roundworms belong to which phylum? They belong to Aschelminthes. Okay. Aschelminthes. And they have the organ level system of organization. Okay. Now, the fourth one is comb plates present in ctenophores help in digestion. This is not at all true. Why? Because comb plates, see, first let's understand there are 8 external rows of comb plates, okay? 8 external rows, okay? And it helps them in the locomotion or movement, okay? So, this statement is incorrect. Now, the next one is water vascular system is a characteristics of echinoderms. That is absolutely true. Okay. So this is the most special features of echinoderms, and what does it help in? It helps in a lot of activities such as locomotion, capture and transport of food, as well as respiration. So this is correct. So how many statements were correct? 3 statements: B, C and E. Okay, A and D were not correct. So B, C and E. So option D is correct here. Okay, I hope you understood this. So now we have question number 67, which is from the chapter Human Reproduction, class 12. And the question is, receptors for sperm binding in mammals are present on corona radiata, vitelline membrane, perivitelline space and zona pellucida. So the correct option is option D, that is zona pellucida. So let's have a look at the structure of an ovum. Suppose this is an ovum. Okay. So this ovum is lined by a membrane which is known as vitelline membrane. Okay. Now there is a space outside this vitelline membrane, and this is known as perivitelline space. Okay. Because peri means around and vitelline, since it is around the vitelline membrane, therefore it is known as perivitelline space. Okay. Now the most important layer is around this perivitelline space, which is known as zona pellucida layer. Okay. So this shaded portion represents the zona pellucida layer. Okay. Now why is this layer important? because this is the layer with which the sperms come in contact while the fertilization is going on. Okay. Now even around this zona pellucida layer is the layer containing cells of radiata, corona radiata. Okay. These are radially arranged cells. Okay. And these are the cells of membrana granulosa and they are known as corona radiata. Okay. So we know that zona pellucida is the correct option here. Okay. Okay, so question number 68 is from the chapter Animal Kingdom class 11 and here we have to match correct features or characteristics with the phylums to which they belong. Okay, so the first one is metamerism and this is the feature of which phylum? Annelida. Okay, what happens here? Suppose this is the body of Annelida. So the body is divided externally and internally into linear segments. Okay, and these linear segments are known as metameres. And this characteristic is known as metamerism. Okay, so the correct match for A will be number 3, right? This one. What is next? Next characteristic is canal system. And this is a characteristics of which phylum? Porifera. What happens here? The body of Porifera has two types of pores. One type of pore is smaller pores all over the body known as ostia. Okay, and water enters into the body through this ostia, then enters into the central cavity known as spongocoel, and then water moves out through the larger pore known as osculum. Okay, so this system is the feature of porifera. So the correct match for B will be number 4, right? Now, number C is comb plates. Comb plates are the characteristics of ctenophora. Okay, ctenophores have 8 external rows of ciliated comb plates which help in locomotion. Okay, so the correct match for C is number 2. And the last one is cnidoblasts. Cnidoblasts are the special type of cells which are found in the phylum Coelenterata. Okay, so the correct match for D is number 1. So which of the given options correctly fit this? Number C, right? So option number C is the correct option here. Now next is question 69. It is from the chapter Chemical Coordination and Integration, class 11. And the question is, erythropoietin hormone which stimulates RBC formation is produced by alpha cells of pancreas, the cells of rostral adenohypophysis, the cells of bone marrow and juxtaglomerular cells of the kidney. So which is the correct option? The correct answer here is option number D, that is juxtaglomerular cells of kidney. Okay, these produce erythropoietin and this hormone stimulates the RBC formation. Okay. Now, what are the other options? Alpha cells of pancreas. What is produced by alpha cells of pancreas? It produces the hormone glucagon. Okay. Number B is the cells of rostral adenohypophysis. Okay. So these cells synthesize hormones of anterior lobe of pituitary. Okay. So anterior lobe of pituitary gland. Okay. Now, the third one is the cells of bone marrow. Now, what is the cell of bone marrow responsible for? It is responsible for the formation of formed elements. Okay. So, it is a direct question that you have to remember. So question number 70 is from the chapter Reproductive Health, class 12, and the question is venereal diseases can spread through. So we have to answer what are the means through which these diseases can be transmitted. What are venereal diseases? These are sexually transmitted diseases. Okay, that means they are transmitted through the sexual means. Other than that, these are also transmitted through any methods that involve the transfer of body fluids from one person to another. Okay, so sterile needles, yes, these are the means through which the liquid can be transmitted. Okay, so this is correct. The next one is transfusion of blood from the infected person. Again, blood is a body fluid, so if this happens, then these will be transmitted. Okay, then the next one is infected mother to fetus. Again, it is true, it can cause the transmission of sexually transmitted diseases. Next is kissing. Kissing is a physical process, right? It does not include the transfer of body fluids. So this is not the correct answer. And the last one is inheritance. So inheritance is also not responsible for the transmission of venereal diseases. So the only true ones are use A, B and C. Okay. So option A is the correct one. Now question number 71 is from the chapter Structural Organization in Animals, class 11, and the question is which of the following characteristics is incorrect with respect to cockroach? Okay, so the first statement is a ring of gastric ceca is present at the junction of midgut and hindgut. What is a gut? Gut is the main digestive organ in the insects. Okay, now in case of cockroach, the gut is composed of three compartments. The first one is foregut, second one is midgut, and the last compartment is hindgut. Okay. Now at the junction of foregut and midgut, you will find gastric ceca or hepatic ceca, which is responsible for the secretion of digestive juices. Okay, whereas at the junction of midgut and hindgut, what is found? Malphigian tubules. Okay, so the first statement is wrong. Why? Because the ring of gastric ceca is present at the junction of foregut and midgut, not at the midgut and hindgut. Okay. Now, the second statement is hypopharynx lies within the cavity enclosed by the mouth parts. Okay, so let's see what are the components of mouth parts in cockroaches. Okay, so mouth parts in cockroach is composed of a labrum or upper lip. Okay, then there is a labium or lower lip. Then there is a pair of mandibles. Okay, that means two mandibles, one here and one mandible here. Okay, then there is a pair of maxillae, one maxilla here, one maxilla here. Okay, so now within the cavity that is enclosed by the mouth parts lies the hypopharynx, and it acts like a tongue. Okay, it acts just like tongue. Okay, so this statement is true. Okay, now the third statement is in females, seventh to ninth sterna together form a genital pouch. Okay, so this is also a correct statement. The abdomen in both males and females consist of how many total sterna? There are total 10 segments. Okay, 10 segments are there. Now out of that, 7th, 8th and ninth, they together form the genital pouch. Okay, so this is a correct statement. Now option D is 10th abdominal segment in both sexes bears a pair of anal cerci. So this is also true. Okay, the 10th one in both the sexes bears a pair of anal cerci. What are anal cerci? They are a pair of jointed filamentous structures. Okay, so here which will be the incorrect option? Number A. Okay, this will be the answer. Now question number 72. It is from the chapter Animal Kingdom, class 11. And here we have to match the animals with the common names given to them. Okay, so the first one is Physalia. Which phylum does Physalia belong to? It belongs to Coelenterata, or you can say Cnidaria. And the common name given to the Physalia is Portuguese man of war. So the correct match for number A will be 2. Okay, Portuguese man of war. Second is Limulus. Limulus is also known as king crab. Okay, and it is considered as a living fossil. So the correct match for B will be number 3. Okay, so living fossil here. The third one is ankylostoma. What is an ankylostoma? It is a type of round worm, and the name given to it is hookworm. Okay, so the correct match for C will be number 4. And the last one is Pinctada. Which phylum does Pinctada belong to? It belongs to the phylum Mollusca. And the common name given to it is pearl oyster. So the correct match for D will be number 1. Okay. So out of the given 4 options, which one does fit this correctly? Is the number C, right? So this is the correct option here. Now come to question number 73, and it is from the chapter Animal Kingdom, class 11. Question is, which one of the following organisms bears hollow and pneumatic long bones? So it is very simple question. You just have to know that which class of organisms have these characteristics of hollow and pneumatic long bones. So these are the characteristics of which class? Class Aves. Okay, that means all the birds will have such bones, right? Now let's see the option. First one is nephron. So nephron means vultures. Okay, so vulture is a bird, therefore it will have such bone, therefore A will be the correct option. Let's see what are the other options. Second one is hemidactylus. What is hemidactylus? Wall lizard. And which class does this belong to? It belongs to the class Reptilia, right? Now the third one is Macropus. Macropus means kangaroo. And the fourth one is Ornithorhynchus. What is Ornithorhynchus? It is platypus. And kangaroo and platypus both belong to which class? Mammalia. Therefore, these will not be the correct option because these are the characteristics of class Aves only. Okay, so A is the correct option. Now question number 74 is from the chapter Cell Cycle and Cell Division, class 11, and the question is, the centriole undergoes duplication during S phase, prophase, metaphase and G2 phase, and the correct option is option A, that is S phase. Okay, so we know that cell cycle is composed of two main phases, the interphase and the mitosis, right? So we know that the interphase lasts for about 95% of the total time, right? And it is further divided into 3 phases. First one is G1 phase, second one is S phase, and the third one is G2 phase, where G1 marks the end of mitosis, right? And the initiation of DNA replication, whereas the S phase marks the actual DNA replication. And in case of animal cells, what happens? The centriole duplicates in the cytoplasm. So duplication of centrioles take place. Then comes the G2 phase. Here some important organelles such as mitochondria, chloroplast and Golgi bodies takes place. Okay. Other than that, one of the important things that happens during G2 phase is synthesis of protein, okay, such as tubulin. So these are the features of G2 phase. Now, mitosis, how many phases is mitosis divided into? 4 phases. The first one is prophase, the second one is metaphase, third one is anaphase, and the last one is telophase, right? So what happens during the prophase? During this, the condensation of chromatin starts, okay? So the chromatin start condensing. During the metaphase, what happens? The chromosomes align themselves at the equator, okay? And these are attached with the spindle fibers on both the poles, right? What happens during anaphase? Here the centromeres split and the chromatids separate. Okay. At the same time, chromatids start moving to the opposite poles with the help of spindle fibers. Okay. And the last one is telophase, during which what happens? The chromosomes cluster at the opposite spindle poles and their identity is lost as discrete elements. Okay. So here the identity is lost and the nuclear envelope assembles around the chromosome clusters. Okay. So you understood what happens during the other phases mentioned in the options. Okay. Now the next one is question number 75, which is from the chapter Biotechnology, Principles and Processes, class 12. And the question is, during the process of gene amplification using PCR, if very high temperature is not maintained in the beginning, then which of the following steps of PCR will be affected first? And the options are annealing, extension, denaturation and ligation. So the correct option is option C, that is denaturation. Why? Because this is the first step of PCR and it involves the application of high temperature. Okay. So what is PCR? PCR stands for polymerase chain reaction, right? And it consists of 3 steps. The first one is denaturation. What happens during denaturation? A very high temperature of about 94 degrees Celsius is applied, due to which the DNA strands are separated and the DNA gets denatured. Okay. The second step is annealing. During this process, a comparatively lower temperature, about 50 degree to 60 degrees Celsius is applied, right? And a primer is annealed at the 3' end of the template strand. Okay. Now this primer is very important because in the last step, that is extension, this primer is extended. Okay. And this is extended with the help of DNA polymerase enzyme using the nucleotides provided in the reaction and the genomic DNA as template. Okay, so this is the process of DNA replication. Now if this DNA replication is repeated many times, the segment of DNA can be amplified to approximately billion times. Okay, so now we understand about the annealing and extension as well. What is ligation? Ligation is the process of joining of DNA fragments with the help of which enzyme? DNA ligase. Okay. So I hope you understood this. Next question is from the chapter Strategies for Enhancement in Food Production, class 12. And the question is, which of the following is not an objective of biofortification in crops? So what is biofortification? It is the process of breeding crops with higher level of vitamins and minerals or higher proteins and healthier fats. Okay. So let's write it down. Breeding crops with higher levels of vitamins and minerals, and higher proteins and healthier fat. Okay. Why is it done? It is the most practical means to improve the public health, okay, and it improves the overall nutritional quality of the food, right? Now, the main purpose for this is to improve the protein content and quality, then to improve the vitamin content of the food, micronutrients and mineral content, and also oil content and quality, okay. But what is not included is disease resistance, okay. So option B is not an objective of the biofortification. So this will be the correct option here. Now question number 77 is from the chapter Environmental Issues, class 12, and the question is Dobson units are used to measure thickness of CFCs, stratosphere, ozone and troposphere. So the correct option is option C, that is ozone. Okay, so we know that our atmosphere has several layers, right? The lowermost layer of the atmosphere is troposphere, right? And the layer above the troposphere is known as stratosphere. What is the main characteristic of stratosphere? It is that it contains the very important ozone. Okay. Why is ozone important? Because it protects us from harmful ultraviolet rays of the sun, right? Now, the thickness of this ozone in a column of air from ground to the top is measured in which unit? In terms of Dobson unit. Okay. So, Dobson unit is the unit for the measurement of ozone thickness. Okay. Here, just remember 1 Dobson unit is equal to 1 parts per billion. Okay. Now, what is CFCs? CFC is an ozone depleting substance, and these are released from our household items such as refrigerators, aerosol sprays, air conditioners, etc. Okay. So, got this? Now question number 78 is from the chapter Digestion and Absorption, class 11, and the question is sphincter of Oddi is present at option A ileocecal junction, option B junction of hepatopancreatic duct and duodenum, option C gastroesophageal junction, and option D junction of jejunum and duodenum. So the correct option is option B, that is junction of hepatopancreatic duct and duodenum. Okay, so let's try to understand what is hepatopancreatic duct and what is duodenum. Okay, so we know that the bile that is secreted by the hepatic cells passes through the hepatic ducts and it is stored in and concentrated in a thin muscular sac known as gallbladder. Okay, so suppose this is your gallbladder. So here you will find the duct from the gallbladder, right? And here there are ducts from the liver. Now the ducts from the liver and the ducts from the gallbladder make up a common bile duct. Okay, here common bile duct or simply bile duct. Okay, now here is an important organ known as pancreas, and at this position you will find the pancreatic duct. So here you have common bile duct and here you have pancreatic duct. Okay, these two will open together in the first part of the small intestine, that is duodenum. Okay, so this common bile duct and the pancreatic duct will open together as hepatopancreatic duct in the duodenum, and it will be guarded by a sphincter known as sphincter of Oddi. Okay. So where is sphincter of Oddi present? At the junction of hepatopancreatic duct and the duodenum, right? What is duodenum? It is the first part of small intestine. So the small intestine has three parts. First part is duodenum. Second part is jejunum. And the third and the last part is ileum, right? Similarly, large intestine also has three parts. The first part is cecum. Second part is colon. And the third part is rectum. So the food material passes through the duodenum, then jejunum, then ileum, and then cecum, then colon, and lastly at rectum. Okay. So the junction present between the ileum and the cecum is ileocecal junction. Okay. This the first option. Okay. An ileocecal valve is present at this junction to prevent the backflow of the fecal matter into the ileum. Okay. What is gastroesophageal junction? The junction present between the esophagus pipe and the stomach. Okay. So the sphincter that regulates the opening of esophagus into the stomach is known as gastroesophageal sphincter. Okay. So I hope this question is clear to you. So question number 79 is from the chapter Breathing and Exchange of Gases, class 11. And the question is, select the favorable conditions required for the formation of oxyhemoglobin at the alveoli. Okay. So four possible conditions are given in the options and we have to find out the most appropriate condition for the formation of oxyhemoglobin. Right. Now, what is hemoglobin? We all know that hemoglobin is a red colored iron containing pigment that is present in our RBCs, right? And it helps in transporting oxygen from our lungs to the peripheral tissues. And what is the use of this oxygen? Here in the tissues, it is used to carry out the cellular metabolism, right? Which kind of metabolism? Aerobic metabolism. Now, this process starts after the oxygen binds with the hemoglobin, forming a complex known as oxyhemoglobin, right? So, this oxyhemoglobin moves from the lungs to the tissues. Okay. Now there are primarily four things that affect the formation of oxyhemoglobin, right? First one is partial pressure of oxygen, then partial pressure of carbon dioxide, then hydrogen ion concentration and temperature, right? Now what is the appropriate condition for the formation of oxyhemoglobin? The most favorable condition is when the partial pressure of oxygen is high whereas all the other things are low. That means lower partial pressure of carbon dioxide, lower hydrogen ion concentration and lower temperature. Okay. And this is the condition that is found in the alveoli. Therefore, oxyhemoglobin is formed at the alveoli. Okay. So which of the options correctly fit this? Option A, okay, high partial pressure of oxygen, low carbon dioxide, low hydrogen ion concentration and lower temperature. Now, what will happen if the condition is reversed? That means then the partial pressure of oxygen will be low and other things will be high. Then in that case, the oxyhemoglobin will be dissociated. So, dissociation of oxyhemoglobin will take place, okay. So this option B is the proper condition for the dissociation of oxyhemoglobin, right? Now this question is from the chapter Biomolecules, class 12. And here we have to find the incorrect pair, okay? So on the left hand side we have some substances, different type of substances. And on the right hand side we have examples of these substances, okay? So we have to find out which of them is not the correct example of particular substance, okay? So let's see, the incorrect one is option D. Why? Because ricin is not a drug. What is ricin? Ricin is a type of toxin and it is obtained from the ricinus plant. So this is the incorrect match. What will be the examples of drugs? Some of the examples of drugs can be vinblastine or curcumin. Okay, so these two are the examples of drugs, whereas ricin is the example of toxin. Okay, and all the other are correct match. Okay, for example, concavalin A is a type of lectin, so this is correct. Okay, then codeine is also the correct example of alkaloid. Other than codeine, what is common example of alkaloid? Morphine. Okay, so these two are very common examples of alkaloid. Then the third one, that is aberrin, is also the correct match for toxin because this is a type of toxin. Okay, and it is obtained from which plant? Aberrus. So now we know that option D will be incorrect. The next question is from the chapter Locomotion and Movement, class 11, and the question is, which of the following statements wrongly represents the nature of smooth muscle? Okay, that means which of the given option is not the characteristic of smooth muscle. Okay, so let's first understand what is a muscle. Muscle is a specialized tissue of mesodermal origin, right? That means they arise from the mesoderm of the embryo. Okay, now the muscles are classified into three types based on the locations they are present in. Okay, so the first one is skeletal muscle, the second one is visceral or smooth muscle, and the third one is cardiac muscle. Okay, as you can understand from the name that the skeletal muscle are those muscles which are closely associated with the skeletal components of our body. For example, your limbs. Okay, similarly, cardiac muscles are the muscles of the heart, whereas visceral or smooth muscles are found in the inner organs of our body. Okay, for example, your digestive tract, alimentary canal, your blood vessels, uterus, etc. Okay. Now, skeletal muscles are under our control, right? You can control them as and when required. Therefore, these are also known as voluntary muscles. Okay. The smooth muscle or the visceral muscles are not in our control. Therefore, they are known as involuntary muscle. Similarly, cardiac muscle is also involuntary because we cannot control the muscle of our heart. And one more characteristic of these muscles is that if you try to see the skeletal muscle under the microscope, you will see striations or striped like appearance. Okay, so these are known as striated muscles because of having striations. Okay, cardiac muscles will also show striations, so these are also striated muscles, whereas smooth muscles do not have striations, therefore these are non-striated. Now let's have a look at the options. The first one is that the smooth muscle, we are talking about smooth muscles here, right? So these muscles have no striations. This is a correct statement. Why? Because smooth muscles have no striations. We saw here. Now, second statement is they are involuntary muscles. So this is also a correct statement because we cannot control these muscles. So these are involuntary. Third option is communication among the cells is performed by intercalated discs. This is not a correct statement. This is wrong. Why? Because intercalated discs are found in the heart muscles or cardiac muscles where they help in cell to cell communication. Also, they link the two cells together and define their boundary. Okay. So these are not the feature of smooth muscles. And the last option is these muscles are present in the walls of blood vessels. This is true. Okay, other than blood vessel, they are also found in digestive tract, alimentary canal, etc. Okay, so the only wrongly represented statement is option C. Okay. All right, now move to question number 82, which is from the chapter Biotechnology and its Applications, class 12. And the question is, for effective treatment of the disease, early diagnosis and understanding its pathophysiology is very important. Which of the following molecular diagnostic techniques is very useful for early detection? Okay, so we know that molecular diagnostic techniques are used for early detection of any disease. Okay, and there are some methods which are included in the molecular diagnostic techniques. Some of them are recombinant DNA technology. Then there is PCR or polymerase chain reaction. And then ELISA is also one of the methods used here. The full form of ELISA is enzyme linked immunosorbent assay. It is used to detect the presence of HIV virus in AIDS patients. Okay. Now, this ELISA is based on which technique? It is based on the principle of antigen-antibody interaction. Okay. So, antigen-anti, that means whenever the antigen will enter our body, the body will fight with them by releasing the antibodies, right? So, here we can detect whether there is infection by pathogen by either detecting the presence of antigens or by detecting the antibodies that are synthesized in the body against the pathogens. Okay, so this is your correct option here, right? Let's see what are other options. First one is Western blotting technique. What is Western blotting technique? It is a technique or blotting technique that is used to find the sequence of amino acid. Okay, that means this is used to identify specific amino acid sequences in protein. On the other hand, sudden blotting technique is used to identify specific sequence of DNA. Okay. So these are not related to the molecular diagnostic techniques. Okay. Next is question 83, which is from the chapter Reproductive Health, class 12. And the question is match list 1 and list 2. So here we have two columns. Column 1 includes some important birth control methods, whereas column 2 has mentioned the mechanism that is followed in these methods. Okay. So we have to match them correctly. Number 1 is walls. What are walls? These are the barrier methods of contraception which are used in females. Okay, so these are barrier. These act as barrier in females, and what do? They block the entry of sperm into the cervix. Okay, so they block the sperm entry. Okay, other than walls, what are other methods of barrier contraception? Diaphragms and cervical cap are also included in it. Okay, so any of these can be used as barrier methods of contraception. Okay, so the correct match for A will be number 1, that is entry of sperm through cervix is blocked. Okay, so it is already correctly matched. Now the second one is IUDs. What are IUDs? They stand for intrauterine devices. That means they are placed inside the uterus. Okay, and what do? They help in phagocytosis of the sperm. That means the sperm will get destroyed by these IUDs, okay, or they will be made ineffective to carry out the fertilization, right? So what will be the correct match for B? Number 3, that is phagocytosis of sperms within the uterus. Okay, now the next are vasectomy and tubectomy. Okay, so let's see what is vasectomy. First of all, remember that vasectomy is carried out in males, whereas tubectomy is followed in female. Okay. What is done in case of vasectomy? Here a surgical method is followed where small part of vas deferens is removed or tied up through small incision. Okay, so suppose this is your testis. Okay, so we know that sperms are produced within the testis, and from here they are transported through the vas deferens. Okay, so in order to prevent this transport of sperms, the vas deferens is cut here and tied. Okay, so this way the sperms will not be transported. Okay, whereas in case of tubectomy, similar surgical method is followed, but here what is cut? Fallopian tube is cut. Okay, so suppose this is your ovary, eggs are produced in the ovary, from here they are transported through the fallopian tube. Okay, to prevent this transfer, the fallopian tube is cut and tied up. Okay, so the correct match for vasectomy will be removal of vas deferens, whereas the correct match for tubectomy will be removal of fallopian tube. Okay, so which of the given options correctly fit this? Option B. Okay, so this will be the correct option. Now question number 84 is from the chapter Cell, the Unit of Life, class 11, and the question is the organelles that are included in the endomembrane system are, and here are the given options. We have to find the correct list of organelles. Okay, so these organelles are endoplasmic reticulum, Golgi complex, lysosomes and vacuoles. So option B gives the correct list of organelles that are included in the endomembrane system. Therefore, this is the correct option. Okay, other options are not correct because in some options mitochondria is mentioned, in some options mitochondria as well as ribosomes are mentioned, and both of these are not the part of endomembrane system. Okay, what is mitochondria? It is also known as energy house of the cell, right? And this is a semi autonomous cell organelle, whereas ribosome is which type of organelle? It is non-membranous. Okay, so only option B will be the correct one. Now question number 85 is from the chapter Cell Cycle and Cell Division, class 11, and the question is which stage of meiotic prophase shows terminalization of chiasmata as its distinctive features? Okay, and the options are leptotene, zygotene, diakinesis and pachytene. The correct option out of these is option C, that is diakinesis. Okay, so let's see, we know that meiosis consists of two sequential cycle of cell division, right? First is meiosis 1 and the second is meiosis 2. Okay, now meiosis 1 consists of prophase 1, metaphase 1, anaphase 1 and telophase 1. Similarly, meiosis 2 consists of prophase 2, metaphase 2, anaphase 2 and telophase 2, right? Out of these, prophase 1 of the meiosis 1 is the lengthiest stage of meiosis as well as the more complex than others. Okay, now this prophase 1 is further divided into 5 stages. What are these stages? First one is leptotene, second one is zygotene, third one is pachytene, fourth is diplotene, and the last one is diakinesis, right? What happens during leptotene? Here the chromosomes become visible under the microscope and they are compacted. So this includes compaction of chromosomes, right? Zygotene, what happens during zygotene? Chromosomes pair up by synapses. Okay, sorry, synapses. So synapses takes place during which the chromosomes pair up and it forms what? homologous chromosomes. That means these paired up chromosomes are known as homologous chromosomes. Okay. Now what happened during pachytene? Here crossing over occurs between the non-sister chromatids of the homologous chromosomes. Okay. Crossing over takes place. Okay. Now what happens during the diplotein? In this stage, an X-shaped structure is formed that is known as chiasmata. Okay, so this is formed in this stage, but this chiasmata is terminalized in diakinesis. Okay, so this includes terminalization of chiasmata. Got it? Therefore, the correct option will be option C, that is diakinesis. Fine. Now question 86 is from the chapter Human Reproduction, class 12, and the question is, which of these is not an important component of initiation of parturition in humans? Okay, so what is parturition? See, at the end of gestation, when the fetus is developed completely inside the womb, then it is expelled out, and this process is known as parturition, right? This is not a simple process because a complex neuroendocrine mechanism is involved in controlling it. Okay, and this neuroendocrine mechanism consists of various hormones. Let's see what happens. First of all, there is an increase in the estrogen level. Okay, therefore the overall estrogen and progesterone ratio also increases. So option 1 is an important component of parturition. Okay, but what is asked here, not an important, so we have to see which of them is not an important component. First one is the important component. Then what else happens? Prostaglandins, which stimulate the uterine contractions, are also produced. Okay, so synthesis of prostaglandins is also an important component here. Okay, what does this do? It acts on the myometrium. Okay, acts on myometrium. Then oxytocin is also released. It is the main hormone, also known as birth hormone. Okay, birth hormone, and it is released by maternal pituitary, which brings about strong uterine contractions. Okay, so it is involved in uterine contraction, and you know how important is uterine contraction for the process of parturition, right? The last one is release of prolactin. So this is not an important component. Why? Because prolactin is a lactation hormone. Okay, so this will be useful after the process of parturition. After the baby has come out, it has to be lactated. So this is not the component during parturition or at the initiation of parturition. So this will be the correct option here according to the question. Now question 87 is from the chapter Strategies for Enhancement in Food Production, class 12, and the question is, which of the following is not a step involved in multiple ovulation embryo transfer technology, that is MOET. So what is multiple ovulation embryo transfer technology? It is a program designed for herd improvement. Okay, and what is the specific motive here? Our motive is to improve the chances of successful production of hybrids. Okay. Now, what is done? A cow is administered a hormone. And what does this hormone do? The function of this hormone is to stimulate follicular maturation. Okay. And super ovulation. That means instead of one egg that they yield per cycle, now they will yield more than one eggs. Okay, about 6 to 8 eggs will be produced at a time. Okay, now let's have a look at the options. First option is cow is administered hormone having LH like activity for superovulation. See here what we wanted follicular maturation. Therefore, what kind of hormone we need? FSH like activity. Okay, the hormone with FSH like activity. It stands for follicular stimulating hormone. So this will help in follicular maturation, not the LH like activity. Okay, so it will be FSH like activity, not LH. Okay. Now the next one is cow yields about 6 to 8 eggs at a time. This is absolutely true. Then the third one is cow is fertilized by artificial insemination. Definitely. And the last one is fertilized eggs are transferred to surrogate mothers at 8 to 32 cell stage. That is also true. Okay. After this, the genetic mother is available for another round of superovulation. Okay. So the only statement that is not a step in multiple ovulation embryo transfer technology is option A. Okay. So this will be the correct option here. Now question number 88 is from the chapter Organisms and Populations, class 12, and here we have to match list 1 with the correct option in list 2. So the first one is Allen's rule. What do you mean by Allen's rule? It is related to the adaptation of animals in cold climates. Okay. So according to this rule, the animals that are adapted to cold climate have compact body parts. For example, certain body parts will be shorter and thicker so as to minimize the heat loss. Okay. And why is it important? It is important so that the animal can retain some amount of heat in its body in order to survive in such a cold condition. Okay. And one of the examples is polar seal. This polar seal has shorter ears and limbs in order to reduce the heat loss. Okay. And this is in accordance with the Allen's rule. Therefore, the correct match for A will be number 4. Right. Now, second is physiological adaptation. And one of the most common examples of physiological adaptation is that of kangaroo rat. Where does kangaroo rat lives? It lives in the hot North American desert. So what kind of environment a desert has? Very hot condition, right? So what does the kangaroo rat do? See, there is a scarcity of water in desert, right? So even in the absence of any external source of water, this kangaroo rat is capable of meeting its need by internal fat oxidation. Okay. How does this help? By internal fat oxidation, water is released as byproduct. Okay, that can be used by the animal. Okay. Not only this, kangaroo rat is also capable of concentrating its urine. So it can concentrate its urine and reduce the loss of water through excretion, right? So the correct match for B will be number 1, that is kangaroo rat. Okay. Now the third is behavioral adaptation. Why is this required? See, there are some animals which lack this physiological adaptation. Okay. So they use the behavioral means to adapt in their environment. Okay. For example, desert lizard. What does desert lizard do? What happens when the temperature decreases? What does it do? It basks in the sun. Okay. To increase the temperature. Okay. And absorb the heat. Okay. And when the temperature increases, what does it do? It goes to shade. So this way it maintains a constant body temperature behaviorally. Okay. So the correct match for C will be number 2. And the last one is biochemical adaptation. The match for this is marine fish at depth. So marine fishes live at very depth of the oceans or seas. Okay. So the pressure is very high. Now in order to cope up with that pressure, they use biochemical adaptation. Okay. So out of the given options, which will be the correct fit for this? It will be option C. Therefore, this will be the answer. Now moving to question number 89, it is again from the chapter Organisms and Populations, class 12. And here we have an assertion and reason kind of question. Okay, so the assertion here is a person goes to high altitude and experiences altitude sickness with symptoms like breathing difficulty and heart palpitations. So this is absolutely true, because as you move to higher altitudes, you will experience a kind of sickness, and since this is due to the change in altitude, this is known as altitude sickness. Okay, and the symptoms are breathing difficulty and heart palpitations along with nausea and fatigue. Okay, so these are also experienced. Now what is the reason? The reason is, here it is mentioned that due to low atmospheric pressure at high altitude, the body does not get sufficient oxygen. So this reason is also correct. Okay, because as you move up, the oxygen amount of oxygen reduces. Okay, therefore you do not get enough oxygen, so you experience these kind of sickness. Okay, so here both assertion and reason are true, and reason is the correct explanation of assertion. Okay, therefore the option A will be the correct one. Okay, but note that with time, like gradually, you will get used to it and you will start getting enough oxygen. How? Because the body compensates the low oxygen availability by increasing the red blood cells, decreasing the binding affinity of hemoglobin, and by increasing the breathing rates. Okay, so it is for a short period that you experience this kind of sickness. Alright, now move to question 90. It is from the chapter Biomolecules, class 11. And here we are given a list of statements with reference to lipids. And we have to find out how many of them are correct. So the first statement is lipids having only single bonds are called unsaturated fatty acids. So this is a false statement. Why? Because these are known as saturated fatty acids, not unsaturated. Okay. For a fatty acid to be unsaturated, it needs to have one or more double bonds. Okay, so this is false. Now the next is lecithin is a phospholipid. This is a correct statement. It is a phospholipid and it is found in our cell membrane. Right. Third statement is trihydroxypropane is a glycerol. This is also a correct statement. What is glycerol? It consists of 3 carbon atoms and each carbon atoms bears a hydroxyl group, right? So since 3 hydroxyl groups are there and 3 carbons are there, this is also known as trihydroxypropane. Okay, so this is also a correct statement. The fourth statement is palmitic acid has 20 carbon atoms including carboxyl carbon. This is not true. Why? Because palmitic acid has 16 carbon atoms, okay, including the carboxyl carbon. And the last statement is arachidonic acid has 16 carbon atoms. This is also not true. Why? Because this acid has 20 carbon atoms, okay. So how many are correct? Only 2, B and C. Therefore, the correct option will be C here. Okay. Now come to question 91. It is from the chapter Locomotion and Movement, class 11. And here we have to match list 1 with the correct option in list 2. So the first one is scapula. What do you mean by scapula? It is also known as shoulder blade. That means it is located somewhere near the shoulder, right? And it is a triangular flat bone of the pectoral girdle. Okay, so I am drawing a rough diagram of your rib cage here. Okay, your rib cage is something like this. So here you will have your collarbone or clavicle. Okay, and below this you will find a triangular shaped bone on the dorsal part. Okay, that means on the back portion, which is known as scapula. Okay, so where is it located? On the dorsal part of the thorax. Okay, so the correct match for A will be number 4. So option D has this. Match for A is 4 here. Okay, now the second one is cranium. Where is cranium located? It is located in our skull. Okay. Suppose this is your skull. So cranium consists of flat bones which are fused end to end like this. Okay. So here these small bones are fused with the help of dense fibrous connective tissues. Okay. Fibrous connective tissues. Okay, and these joints in the cranium do not allow any movement. Okay, can you move these joints? No, right? So no movement is there, and these are known as fibrous joints. Okay, so the correct match for B will be number 3. Okay, so here B is matched with 3, right? Now the third one is sternum. Here look at this diagram. You have this bone at the front portion, right? This is your sternum. Okay, scapula was present at the back and sternum is located at the front. Okay, this is also known as breastbone. Okay, because it is located at the front side of your body, that is at the breast. Okay, so it is a flat bone on the ventral midline of the thorax. Ventral means front portion. Okay, and it is present at the midline. Okay, so the correct match for sternum will be flat bone, number 2. Okay, and the last is vertebral column. So the match for vertebral column is the only left option, that is cartilaginous joints. You know, your vertebral column looks something like this and it is composed of 26 serially arranged units known as vertebra, right? So each one of them are known as vertebra, right? And the joints between the adjacent vertebra and the vertebral column are example of cartilaginous joints. Okay, as these are joints with the help of cartilage. So cartilage is used in joining them. So the correct match for D will be 1 and the option that is correct is option D here. Okay. Question 92 is from the chapter Structural Organization in Animals, class 11. And the question is, identify the types of cell junctions that help to stop leakage of the substances across a tissue and facilitation of communication with neighboring cells via rapid transfer of ions and molecules. So how many types of cell junctions are there? Total 3 types of cell junctions are there. Okay. First one is your tight junction. Second one is your adhering junction. And the last one is your gap junction. Okay. Now what does tight junction do? Tight junction stops leakage of the substances from leaking across a tissue. So it stops leakage. Okay. So for the first part, the answer will be your tight junction. Okay. Now what does adhering junction do? It cements and keep the neighboring cells together. Okay. Cements, that means attaches two cells together. Okay. The neighboring cells together. That means it acts as glue. Okay. To connect the cells. And what is the role of gap junction? It helps in communication. Okay, so it is also known as communication junction, right? So it facilitates communication between the cells by connecting the cytoplasm of adjoining cells. Okay, so for second part, the answer will be gap junction. So the correct option will be your option B, that is tight junction and gap junctions respectively. Okay. Now question number 93 from the chapter Molecular Basis of Inheritance, class 12. And here we are given two statements, statement 1 and statement 2. And we have to find out if one or both of them are correct. Okay. So the first statement is the codon AUG codes for methionine and phenylalanine. So the first part is true because it codes for methionine, but it does not code for phenylalanine. So overall the statement is not true. Okay. So here you should remember that AUG has dual functions. First, it codes for methionine. Now, additionally, it also acts as initiator codon, okay, during the process of translation, okay. So the translation starts by this codon, okay. So the first statement is incorrect. But what is the codon used for phenylalanine? Two codons are used for phenylalanine. These are UUU and UUC. Okay, so also remember this thing. Now the second statement says AAA and AAG both codons code for amino acid lysine. Okay, so this is a true statement. Okay, because both of them are the right codon for lysine. Okay, so out of the given options, option D will be correct because statement 1 is incorrect but statement 2 is true. Okay. Now, question number 94 is from the chapter Human Reproduction, class 12, and the question is, which of the following secretes the hormone relaxin during the later phase of pregnancy? And the options are Graafian follicle, corpus luteum, fetus, and uterus. And the correct option is option B, that is corpus luteum. Okay, what is corpus luteum? It is the ruptured Graafian follicle. That means when the Graafian follicle gets ruptured, it leads to the formation of corpus luteum. Okay, and this corpus luteum has endocrine function. That means it secretes various hormones. One of them is relaxin, which is produced during the later phase of pregnancy. Okay, other than that, this corpus luteum also secretes large amount of progesterone during the earlier phase, okay, which is essential for the maintenance of endometrium. Okay, why is it required to maintain this endometrium? Because this endometrium will help in implantation of the fertilized ovum. Okay, fertilized ovum. So now we know that corpus luteum is essential for the pregnancy. Next question is from the chapter Structural Organization in Animals, class 11. And here we are given some statements about prostomium of earthworm, right? And we have to choose the correct answer from the options given here. So first of all, we should know that an earthworm belongs to which family? It belongs to the family Annelida, okay? And one of the characteristic features of annelids is the presence of linear series of segments, okay, known as metameres. And this characteristic is known as metamerism, okay. But at the anterior portion, it has a mouth along with a prostomium, okay. So suppose this is your mouth opening, this is your mouth opening, right? So this is the prostomium. That means it acts as a covering for mouth, right? And then starts the segments of its body, okay. The first segment is known as peristomium, okay. Now come to the statements here. First statement is it serves as a covering for mouth. So this is true. We saw here it is present just above the mouth opening. So it acts as a covering for mouth. So this is a correct statement. Second statement says it helps to open cracks into the soil into which it can crawl. It is also a correct statement. Okay, since this is the anterior most portion, so it helps the earthworm to crawl into the soil. Okay. Now, the next statement is it is one of the sensory structures. It is also true because receptors are present on the prostomium. Okay. Therefore, it is a sensory structure. Now, the last statement is it is the first body segment. So, this is not true because here we saw, see, prostomium and mouth opening are a separate portion. Okay. After that starts the segments. First segment is peristomium, not prostonium. So do not confuse between them. So this is not true. So only options A, B and C are correct, right? So this will be your correct answer. Question number 96 is from the chapter Molecular Basis of Inheritance, class 12. And the question is, which of the following statements about histones is wrong? Okay, so we have to find out the incorrect statement. First one is histones are organized to form a unit of 8 molecules. So this is a correct statement. Okay, histone consists of 5 types of molecules: H1, H2A, H2B, H3 and H4. Okay, out of which 4 occur in pair. These two and these two occur in pair to form histone octamer. That means a unit of 8 molecules. Okay, let's see how. Suppose this is your H2A and this is your H2B. Okay, similarly one more such pair will exist, H2A and H2B. Okay, so how many molecules? 1, 2, 3, 4. So these two form 2 pairs. Okay, similarly H3 and H4 will form pairs together. How many pairs? 1 pair here and 1 pair here. Okay, so H3 and H4 again. So how many total molecules? 1, 2, 3, 4, 5, 6, 7, 8. Okay, so these 8 will form a unit of 8 molecules, that is histone octamer. Okay, now the next statement is the pH of histones is slightly acidic. This is not true because the histone molecules are rich in basic amino acids, lysines and arginines. Okay, so these are basic amino acids. So statement C is true that it consists of lysine and arginine, but statement B is false because these are basic in nature. Therefore the pH of histone will be basic. Okay, not acidic. Now the last statement is histones carry positive charge in the side chain. This is also correct. Why? Because histone has to remain in association with negatively charged DNA molecule, right? You know that DNA is negatively charged. Okay, so in order to remain intact with this negatively charged molecule, it has to be positively charged. Okay, therefore it carries positive charge in the side chains. Okay, so the only wrong statement is statement B. Question number 97. It is from the chapter Locomotion and Movement, class 11. And here some events are mentioned and we have to find out which of these occur during muscular contraction. Okay, so let's first understand the basic structure of muscle fibers. So we know that a muscle fiber consists of two types of proteins. First one is actin protein and the second one is myosin. Now when observed under the microscope, these muscle fibers represent actin as light bands because these are thinner as compared to the myosin, whereas myosin are observed as dark bands because they are thicker as compared to the actin. Okay. Now light bands of the actin are also known as isotropic band or I band in short, whereas dark band is also known as anisotropic band or A band in short. Okay. Now how are these proteins arranged? Both the proteins are arranged as rod-like structures parallel to each other such that the I band or the thin filament overlaps the A band or the thick filaments. Okay. So can you see here that the I bands are overlapping the A band. Okay. Now there is an elastic fiber known as Z line which bisects these I bands. Okay. So all the I bands will be bisected by the Z line. Now the portion between two consecutive Z line is known as the sarcomere and it represents the functional unit of contraction. Okay. Now can you observe one more thing, that even though the I band is overlapping the A band, there is a portion in between that is not overlapped, this one, okay. So this is known as H zone, which is not overlapped by the I band, okay. But as soon as the contraction starts, what happens? We know that myosin has a globular head. So suppose this is a myosin and this is the actin, okay. So the myosin hydrolyzes the ATP molecule and forms a cross bridge with actin, okay. Due to this, the I band start moving towards the center. Now along with I band, the Z line also starts moving towards the center, okay. So the length of I band is reduced, and ultimately what happens? A point comes when the I bands completely overlap the A band, okay. Can you see here? The I band has completely overlapped the A band and no space is left, okay. So H zone here disappeared. No H zone is present. So H zone disappeared here, okay. Now, at last what happens? The myosin releases ADP plus inorganic phosphate and goes back to its relaxed state to start the process again, okay. Now, have a look at the options here. The first one is H zone disappears during muscular contraction. So this is true. We saw here that H zone disappeared. So this is a correct statement. Second is A band widens. So could you see any change in A band? No, right? I band got reduced, but there was no effect on A band. It remains at the same location and the length also remained the same. Okay, so this is false. There was no effect on A band. Third is I band reduces in width. So this is true. We saw here, right? Now, the option D is myosin hydrolyzes ADP, releasing it hydrolyzes ATP, releasing the ADP and phosphate. So this is also true. Okay. And the last one is Z lines attached to actins are pulled inwards. This is also true. We saw now that Z lines started pulling inwards. So this is also correct. So only statement B was incorrect. Rest were correct. That means A, C, D and E were correct. Okay. So option A will be the correct answer. Question number 98 is from the chapter Biotechnology and its Applications, class 12. And the question is, the adenosine deaminase deficiency results into, and the options are dysfunction of immune system, Parkinson's disease, digestive disorder, and Addison's disease. So the correct option is option A. So this is a very direct question. We just have to remember that adenosine deaminase, that is ADA, is responsible for the proper functioning of immune system. Therefore, its deficiency will result into the dysfunction of immune system. Okay. Therefore, option A will be the correct answer here. Now, let's have a look at other options as well. Option B is Parkinson's disease. So, what is Parkinson's disease? It is a long-term degenerative disorder of the central nervous system. Okay. And what is digestive disorder? It should be very clear to you. Okay, what is affected in case of digestive disorder are gastrointestinal tract, right, and all the associated glands. Okay, so it will result into the digestive problems. The last one is Addison's disease. So what is Addison's disease? It is caused due to the hyposecretion, that is the secretion in lower amount, okay, of the hormones of adrenal cortex, okay, hormones of adrenal cortex. So I hope this is clear to you. Question number 99 is from the chapter Evolution, class 12, and here we have to match list 1 with the correct option in list 2. So the first one is adaptive radiation. What do you mean by adaptive radiation? It is an evolutionary process by which many species evolve from the same species. Okay, that means different species originate from one species in any given area. Okay, so Darwin's finches is one of the best examples of adaptive radiation. Finches are nothing but small birds that were observed by Darwin in the Galapagos Island. Okay, so during his journey to Galapagos Island, Darwin observed a variety of small black birds, which he later called finches, and he realized that all of them evolved on the island itself and from the same species. Okay, suppose the original species has a particular type of beak, then the species that originated from it had different types of beaks, and they led to the formation of different species overall. Okay, so this is the adaptive radiation, right? Now next is convergent evolution. So here we should understand the meaning of convergent and divergent. There is just a basic difference between them. Convergent means what? Coming from different points but meeting at a same point, or converging at the same point. And divergent is just the opposite. That means starting from the same point but diverging to different points. Okay. So in the context of evolution, what does it mean? Convergent evolution is the one where the structures are anatomically different. Okay, that means they have different ancestry, but yet they have same function. Okay, for example, wings of butterfly and bird. You know, the wings have different structures, or they are anatomically different, but their function is same, that is flying, right? So this is the example of convergent evolution. Now, divergence is just the opposite. That means they have the common ancestry, but their function is different. Okay. So in spite of having same structure or common ancestry, they will perform different functions. For example, bones of forelimbs in man and whale. So these are anatomically same, but we have different functions, right? Whale does not perform the same function as we do. Therefore, this is the example of divergent evolution. Okay, understood? And the last one is evolution by anthropogenic action. Anthropogenic means by human activities. Okay, so evolution by anthropogenic action means evolution due to human interference. For example, antibiotic resistant microbes, herbicides resistant varieties, or pesticide resistant varieties. That means excessive use of these chemicals had led to a resistant variety of organisms. Okay, therefore this is the example of option D. Okay, so out of the given options, which will be the correct match? It will be option A. Okay. And the last question is from the chapter Human Health and Disease, class 12. And we have to match the names of diseases in list 1 with the causative organisms in list 2. Okay. So the first disease that is mentioned here is filariasis. Okay. And it is caused by filarial worm. What is the name of this worm? The name is Wuchereria bancrofti. Okay. Wuchereria bancrofti. So the correct match for A will be number 3. Okay. Now the second disease that is mentioned here is amoebiasis. And it is caused by a protozoan parasite, or you can say simply protozoa, with the name Entamoeba histolytica. Okay. Entamoeba histolytica. And where are these organisms found? These are found in large intestine in humans. Okay. So the correct match for amoebiasis will be your Entamoeba histolytica, that is number 4. Okay. So here you will see this option is fitting in this explanation, right? Now the third is pneumonia. So you might be aware that pneumonia is caused by some bacteria. Okay. And the main bacteria are, first is Streptococcus pneumoniae. Okay. And the second bacteria that can cause pneumonia is Haemophilus influenzae. Okay. Haemophilus influenzae. So this Haemophilus influenzae is mentioned here. So this will be the correct match for number C, that is pneumonia, right? And the last one is ringworm. So the ringworm is caused by a which kind of organism? fungi. Okay, and the fungi can belong to any of these genera. First one is Microsporum. Second one is Trichophyton. And the third one is Epidermophyton. So the fungi belonging to any of these three genera can cause the ringworm. Okay. So here Trichophyton is mentioned. So this will be the correct match for number D. Okay. So out of the given option B correctly fits this. So this will be the answer.